Welcome to Inside AI, a podcast from the EPFL AI Center. I'm your host, Marcel Salate, professor and co-director of the EPFL AI Center. Each episode brings you fascinating discussions with brilliant minds from EPFL and across the global AI community. From groundbreaking algorithms to ethical considerations, from technical innovations to real-world applications, we explore the technology that's transforming our world. Whether you're an AI researcher, industry professional, or simply curious about the technology that's shaping our future, join us as we venture inside AI. Today I'm speaking with Stef van Grieken, the CEO and co-founder of Cradle. Stef grew up in what the Dutch BBC once called the shittiest village in the Netherlands, which he says gave him plenty of time to spend with computers. After helping build Google's TPU chip in Silicon Valley, Stef had an epiphany. What if we could generate proteins the way we generate images or text? Now his company, which just raised $73 billion in Series B funding, is doing exactly that. Their software recently designed a cancer drug binder that's 8 times better than what's currently on the market, and an intern with no biology background did it in a couple of hours. But this conversation goes way beyond pharma. Stef argues that 60% of what we consume, from laundry detergent to cheese to clothing materials, could be made with engineered biology instead of oil, animals, or plants. After spending 7 years in the Bay Area, he moved back to Europe to build Cradle from Switzerland and the Netherlands, and has some sharp observations why he thinks Europe is shooting itself in the foot. We'll discuss one of his viral LinkedIn posts about losing a star machine learning engineer to OpenAI because Dutch visa processes took 8 weeks. I hope you enjoy the conversation. All right, Stef, thanks for coming on the podcast.
Thanks for having me.
I have just introduced you, but it would nevertheless be good to, before we talk about Cradle, which is your baby, to learn a bit more about yourself from you, so to speak, your journey up to the point, which is pretty interesting, and, pretty much up to the point of founding Cradle, and then we can talk about what happened after, following that.
Yeah, sure. So I'm Stef. I grew up in a really small town in the Netherlands called Opijnen. The local Dutch BBC once called it the shittiest village in the Netherlands. So I had a lot of time to spend with computers. I studied industrial engineering and philosophy all the way up in the north in Groningen, and actually didn't start my career in tech, but in politics. So I worked in the European Parliament for about a year as a staffer to one of the members. Kind of realized I wasn't a great politician, and so then pivoted towards more of a tech career. Spent most of my time at Google, initially in search on lots of interesting queries such as medical queries or elections or places where the answer matters and can be somebody's opinion on Reddit, as the first blue link, if you will. Got to spend a real fun amount of time with sort of early machine learning in Google Maps, so the traffic information and sort of figuring out where the traffic jams are and how to distribute that information. And then sort of started my journey more down the deep end. I joined the Android Auto team, which is an application that if you plug your phone into your car, you get your apps like Spotify and Maps and others sort of projected onto your car dashboard. And then got a bit of a, let's say, call from the people that were doing learning methods, because sort of some of the early days in Google Maps were about, the very early days of learning things from large data sets, and said, hey, we need better hardware for machine learning. And so ended up being one of the 2 leads of a TPU. A TPU is a piece of hardware that does really well machine learning workloads, similar to GPUs, but they're in Google data centers, and they do a lot of training and serving of machine learning models. In that context, I got to meet lots of users of large-scale compute, and there were a couple of teams that were applying it to domains that I didn't know a lot about, in particular biology, and I got super excited about that, and sort of that leads us to Cradle.
And just to finalize that, so to speak, you spent most of the time in the US and some of it in Zurich, is that right?
Yeah, correct. So I actually started out in London for about a year, and then me and my wife moved to the Bay Area. I think I spent 6 or 7 years there, and I moved back to Zurich or to Europe really about 5 years ago now.
And then so you see something in biology that is so fascinating that you say, okay, that's it. My time at Google is over. Even though you're probably at one of the, I mean, most interesting places, right? In sort of terms of tech in general. What was that moment? What was your aha moment to sort of say, okay, I have to go and do this?
Yeah, sure. So large language models were sort of just getting started when I was at Google, and so they were used in different contexts, like Google search and figuring out what answer to give, or places like Google Translate to translate a language from A to B. And when you're building hardware in particular, because the timelines are so long, you need to hang out with a lot of researchers because you need to figure out what are the models going to look like 1 or 2 years from now, right? Because you need to be able to train and serve them in production. And so as part of that, I both met some of the folks on the AlphaFold team that were busy doing protein folding. I didn't know what that was at the time, but I also met my co-founder Eli, who was applying very similar techniques to large language models, but instead of training them on human language, he was training them on DNA sequences, right? And he was like, well, it was kind of cool at the time. Google Translate started to learn languages that we didn't have good translations for. So instead of having one model from like German to English, we would have like one model to rule them all. And there were even certain dialects and languages that were forgotten that started to become translatable, which is pretty cool. And so he was like, hey, akin to how we've learned how to translate from A to B, we might be able to learn how organic chemistry works. And he's a organic chemist by training and sort of became computer scientist later. And I was like, why is that helpful? He's like, well, there's so many things that get built with organic chemistry, right? Drugs and materials and foods. And I think similar to how, we've learned human language, we might be able to learn organic chemistry. And then similarly to what you were seeing at the time, like generative picture models or generative coding models that produce code, is like maybe we can generate molecules, right? These protein molecules and, make them super useful for the physical world. That absolutely blew my mind. And so I started digging in there. And then you realize a couple of things, like pharma is like an order of magnitude larger than anything we do online, because as soon as you've taken your first cancer drug, you've effectively paid for all of your ad clicks in your entire life, right? And, the amount of stuff we consume, but also like the planetary impact of a lot of the things that we consume today is so massive, and we could move a lot of that back into using biology. And so I guess, building general intelligence is cool. And that's definitely something I could have stayed at Google for. But learning the interface to biology, to the physical world, I think is way more exciting than making smart computers.
Hmm. Okay. And then, so then you are founding Cradle that was 2021, right?
Yeah.
And so what, to someone who's not in that field, how would you describe what Cradle does?
Yeah, so Cradle is software that people in the research and development departments of pharma companies, food companies, material companies, chemical companies use to engineer proteins. Now, what are proteins? For those that don't have a biology background, proteins are little machines that sit inside of cells. And there's about 22,000 of them in any human body, but they're in plants, they're in all living things. And you can think of them as the little machines. So they can do lots of different things. They can be signals. So if you press a button like GLP-1 for the drugs that are currently very popular for people that want to lose weight, they press a button and that button triggers a certain process which makes you less hungry. Great. So you need to make a molecule that attaches to that, and that's a protein. Or, in your detergents, there are proteins that can break down fats. So people typically don't like fat stains in their clothing. You can try to break them down by putting a lot of water on, but you can also use proteins because they're very good at breaking down fats, right? And so this is formulated in your detergents today, and it's one of the reasons why they keep working at ever lower temperatures, because lots of biological things work at room temperature, right? They have to. And so these are just some examples. And so instead of us, let's say, generating a video or generating a piece of computer code, we generate a sequence that encodes for that specific molecule that people might want to use in some of these applications. So you can think of us a little bit as like Photoshop for organic chemistry.
Hmm. So that's actually super fascinating. I think that point is generally not super well appreciated, maybe in the broader public, where there's sort of this idea that scientists go to the jungle somewhere and find the molecule that, and like those days, I mean, well, I'm not sure they ever existed, but certainly we were looking for things that nature had already produced, right? Perhaps not always in the jungle, though that makes for good stories. But so now basically you're saying, oh, we don't have to constrain ourselves anymore to what nature produces by default. If we understand its language, we can now design those proteins to have certain properties that we desire for whatever.
Yeah, correct. Yeah. So you can do two things. One is you can take something that existed in nature, but maybe doesn't do all the things that you want. So, for example, people like their molecules to be able to deal with a lot of heat, right? That makes their shelf life longer and that kind of stuff. So you can sort of make them more rigid. And those are properties you can engineer. Or to your point, you can come up with things that are new to nature. So where evolution never came up with a reason to have it in the first place. That's still, let's say, more at the frontier, but I do expect that in the next few years, we'll be in a position where we can make molecules that are very far away from sort of the evolutionary manifold, I think is what they call it.
Yeah. Yeah, I guess the evolutionary pressure for not having fat stains on your shirt was relatively low.
Yeah, and maybe very practical things like, we want to break down plastics, right? There hasn't been a lot of time in nature where plastics had to be degraded by biology, right? And so that's a typical example of an area where I think engineered biology could have major impact. But also, most of the medications that we make are kind of new to nature, right? Because if we knew how to cure our own cancer, we wouldn't have cancer drugs, right? And so any pharma company that might be developing a therapeutic typically looks for something that doesn't really exist in biology, or it may have a starting point in biology, but that needs to be engineered for that specific medical application.
Yeah. And I think that point that you mentioned, that it's actually not just about medication, is also something that's not often enough talked about. We'll get back to that. For now, can you just walk us a bit through the process? So, say I'm a customer, I have, interest, again, for whatever reason, in new proteins with certain properties. I heard that Cradle has a great platform. So I get in touch with you. Then what's assuming we get into business, what's happening? What's the process, to the extent that you can talk about it?
No, of course, yeah. I mean, you can see the screenshots on our website, so happy to talk through it. So there are typically two parts to. Let me first contextualize this. So our users are scientists, so they're people that typically have a molecular biology background, and they typically know a lot about the application. So if you're a detergent scientist and you know a lot about stains, then you might start there. Or if you're developing a drug, you probably know a lot about the particular disease or mechanism of what you're trying to target. So you can use Cradle from what they call discovery. So that's like finding that starting point, like that first thing that does what you want to do, all the way to sort of either in the medical case going into clinical research, or in the industrial case going into sort of a scale-up process where you can actually manufacture the thing at some reasonable scale. So in discovery, what do you start with? You can start with two things. One is you can start with a single sequence of something is sort of similar to what you want. So let's take, cheese making as an example. Like enzymes can turn milk into cheese. Maybe you're like, I kind of like that. We know that exists in nature. I know the sequence of that thing, but I want to use it in this context, right? I want to make more of it. I want it to be faster so we can make cheese in, I don't know, a few hours or days instead of maybe a couple of weeks to get to a certain result. Then you start with that sequence, or you can start with, for example, in the medical case, some understanding of what you're trying to target. And so maybe for your audience to explain what most, let's say, drugs are about, they're either vaccine-like, so they kind of float around in your blood and they get attacked by your immune system, and then your immune system kind of learns how to attack that pathogen. or they're more, they try to bind to something specific. So they're more like, for example, an antibody where it grabs onto something and then, your immune system gets activated and will start attacking the specific disease. And in that case, you could also come with a structure, which is like, what is the molecule I'm trying to grab onto look like? And we can design something against that. Then you go into the software, you upload either that starting point or paste in that sequence or whatever set of sequences you have that you like. And then you have to determine what you want from the molecule. And so the way that a scientist would prompt our models is by describing what biologists typically call a TPP, a target product profile. Think of it almost as a requirements set for your molecule. So, in biology, there's lots of things. Let's take, a drug, for example, is you want to bind to the target. You want to bind with a certain strength. And so you can describe, I would like binding affinity to be this number of nanomoles or below, right? Then, there's typically, you want the be able to make enough of it. In biology, you call that expression. And so you can say, hey, I want this many milligrams per liter to be produced in a cell. You might say something like, hey, I want it to be thermally stable at 45 degrees, because if it falls apart as soon as it hits the bloodstream, that's not very helpful, right? And so you can sort of set all of these requirements, and then akin to how you would use in any other generative model, you hit like generate or enter, and then, model starts producing some stuff. Now, in human language, you will typically get an answer in a couple of seconds or minutes. Given how complicated biology is, this can last hours and sometimes even a day to generate typically whatever fits in your experiment. So that's typically 96 or 384 samples, like design sequences that people will then take into their laboratories. Now, every biology team will typically go through several rounds of experimentation until they reach this desired end state at this target product profile. And so we'll stay with them every round as they progress. And so they'll keep uploading their data, change all bunch of parameters, maybe add information that they have in their head, hit generate again, and go to their lab until at some point the R&D department goes to clinical development team and says, here you go, here's a sequence of our new drug. Why did you go test this in a clinical study? Or we'll take it to the people that do scale up in an industrial bio context and say, hey, put this in a large steel tank and make a lot of it, and then go sell it to whomever is the consumer of that ingredient.
Mm-hmm.
Right?
And why does it take long? And of course, we say long here in quotes because it's still super fast compared to any alternative process. But you made that comparison to like a regular large language model, where tokens are predicted. I don't know what the current record is. I think Cerebras has the current record with like 2,000 tokens per second.
You have new records every day. Yeah,
exactly. And you're saying no, this can take hours. So is it because your model is so large, or is it and it needs just a lot of computation, or are there some reasoning steps involved where you constantly trace back and say no, this was wrong, I have to go this way?
Yeah, so let me maybe contrast and share some of the problems that we have with biological models compared to human language models. I think that the biggest issue in biology is that we're in the case of models that operate on sequences, is that we can read sequences. That's problem number one. So if I were to give you a sequence of chymosin, right? You could look at it and be like, I don't know what this is, right? Because we don't know how to program biology as humans. And so the second step that we need to do is we need to sort of translate it to things that humans know how to utter, like how to prompt, if you will. And so, for example, we talked about stability. And so we don't just train on raw sequences, but we train on measurements that we have about biology, because for many sequences, we just don't know what measurements we're going to get. Right? So that's problem number one is we have a ground truth we don't know, right? And we can't read. The second problem is we're very data poor in biology. And so there's only in public databases a few billion protein sequences, whereas let's say in human language, we have lots of text, we have lots of pictures, we have lots of videos that humans made, and they're pretty decent coverage of the things humans would typically do on the internet. Whereas in biology, that's very much not the case. We've only sampled a very small amount of sequences, and then we almost have no experiments because experiments are very expensive, right? So between a couple of hundred up to tens of thousands of dollars for one data point, and it takes 3 months to get one of these data points. And so, and then even worse, if you found something cool, you're typically going to patent it and not put it in public domain. So most of the things that are on the public internet are actually not very useful for these models to train on. So these are some of the contrasts. So why does it take too long or so long, to your point? So for us, the real problem is to figure out, are we in domain or not? Are we hallucinating or not? And so the way that we do that is we have a generator, and the generator comes up with sequences. Now, that is already a quite complicated problem because the sequence is written in 20 letters. So you have 22 to 200th for a small protein in terms of positions that you can alter. That's more than we have atoms in the universe. So you can't like exhaustively search that. So you need to find a way for your generator to be good enough, right? The second thing is then we have a predictor. It's a multi-headed predictor, and it kind of understands these different properties that people want, and they sort of play with each other. So think of it as an artist and an art critic. And so the artist generates maybe 1 billion variants, and then the art critic gets to judge whether they like the new sequences or not. Now, that process of them sort of playing with each other, that takes a long time. And to your point, there are forks and ways to go back and forth there, where that's an online process where decisions get made inside the model. And so, that can, depending on the complexity of what you want, can take quite a lot of time and a lot of machines. Yeah.
Okay. And if I understand correctly, one of the what's special about Cradle also is that you're not just software, right? You also have a lab attached to in your process. Is that right?
Correct. Yeah. So because sort of generating is hard and there aren't a lot of labels, you need to kind of make sure that what you do works in a very broad context, right? If you sell software, it needs to work consistently, not just in a very, let's say, few set of cases. And so the analogy with, for example, self-driving cars is our laboratory is our car, right? And so every time we have a better driver, we go test it out on the car and figure out, did it become a better driver or not? Because we need to test that what we generated actually translates into the real biology, if you will. And so we don't use our lab to make individual products, but it's a way to test, did our new version of our model improve or regress compared to what we had before? So that's one. Second, and this is more of a function of biology, is that the way that we collect data in biology is very localized, right? So if you are making a cancer drug against, epidermal growth factor, right, which is a highly common receptor on many cancers, you're kind of searching a very local neighborhood, right? You're creating a lot of data that's very specific to that problem, which would be akin to you're driving your car around the same block, right? Or the same 2-3 streets a lot. So you learn a lot about, there's like trees over there. There's 3 parking spots there. Mr. John shows up on Monday morning. But as soon as I put that car on the highway, it will crash, right? Because it doesn't know where it is at that. So the second thing that we do, and I think this is also different about biology, is we build these data sets that are specific to a property, but not, but general for lots of our customers. So for example, we always, or typically we want a protein to be thermally stable. So we don't want it to fall apart if the temperature gets too high. You were, the big face of COVID. You probably talked a lot about moving COVID vaccines around in -80 freezers. Typically, people don't like that if they have a molecule that falls apart at room temperature, right? And so, we've collected, I think, something along the lines of 40,000 measurements of when a protein falls apart for very different sequences. And so you can sort of intuit that our model has learned, what does a stable protein look like? And so when it then generates, it makes something that's more likely to be fit for purpose for humans.
All right. Yeah, that's super exciting. So where is this going to go? I mean, if you succeed in the long run, you imagine, 5 years out, 10 years out, where could this take us, right? What could be possible in biotech that isn't today?
Yeah, I mean, that's a really hard question to answer. So maybe let me tell you 2 stories. So one is my co-founder, Jelle. He was one of the first app developers in the world, first employee at Uber, made their app. I don't think Apple would have imagined that would be a thing, but it became possible because, the App Store existed. I think similarly, when I was at Google, we launched TensorFlow, which is software that people use to make machine learning models. At the time, there were only a couple of thousand people that knew how to make a machine learning model, but Google invested in it because they believed that if you could add cognition, right, if you could add intelligence to software applications, that a lot of people would start doing it. Now, I hope something very similar will happen in biology. And so, McKinsey calculated about 60% of the things that we consume today, whether it's your toothpaste, whether it's your hair products, whether it's your food, whether it's the stuff you put in your dishwasher, the nutrients you give to your dog, right? All of these things could be made with biology instead of animals, instead of plants, instead of hydrocarbons like oil, right? But the reason that that's not happening right now is because it's super expensive to develop one of these products, right? So in biopharma, it's typically, tens to hundreds of millions to make one of these. And so you almost need to be a venture firm, by yourself in order to take that risk, right? So what I think will happen, but we're still very much in GPT 0.2 in biology, but what I think will happen is the cost of making one of these molecules will go down from maybe 100 million to, let's say, maybe 2 to 5 million, 10 million. It's hard to say because this is somewhere in the future. But if that's true, and then if the success rates of these things actually making it to the market go up, I think a lot more people will start building with biology, because you can ultimately build better products that way. You can build more sustainable products that way. You can build better medication that way. And so both for human as well as planetary health, that's where I think we're heading. Now, when that will happen, having been at Google, we always set the self-driving cars 5 years out, and it was almost a decade of us seeing that every year. So it will happen someday, but I don't know exactly when. But that's where we're heading, right? So success rates of these molecules will go up, and then the cost to develop them will commoditize ultimately. And I think that's a pretty exciting world.
All right, let's go back to the beginning of the company. So it's always, I think, fascinating, right, to think how all of this starts. So, I mean, you recently announced that you just closed the 73 million Series B. You had, of course, previous fundraising successes. Like, you read this, and of course, you never heard about this as an external person, so everything looks like an overnight success, right? But of course, it never is, right? Somebody at some table somewhere had to say, okay, let's do this. And presumably without any resources, at least in the beginning, or very little. So what was it like for you? What did your first year after founding look like?
Yeah, no, I think that's a great question. I mean, even before founding, it took us about 3 to 4 months to get the team together, right? Because you need deep experts in biology, deep experts in machine learning, but given that we're developing software, also deep experts in application software and design, right? So, first of all is like gather a team and have an interesting enough idea. I think for us, we are quite a privileged team. So, people always point to us and say, oh, it's such a great European success. Well, it's really a bunch of people that were trained in Silicon Valley that came back to Europe and now are building in a very similar way as in the US. And so I don't think any having relationships with venture capitalists in the Bay Area that are more open to taking risk, for example, is an incredible unfair advantage compared to if I had started the company here without that prior background, right? So I wanna also caution this as the let's say outlier, right, and not the default. But anyway, the first year, so we wrote down 5 hypotheses. So one is, can we build the team? Because that's hard, right? And especially in a domain that's as complicated as the one that we're building in. Second is, can we even provide value? And so really goal 1 was to show in the context of a laboratory that we could beat methods that were currently being very commonly used by biologists to engineer these systems. So there were physics-based methods. There were what they call combinatorial libraries. So sort of, I wouldn't call them random, but are we even outperforming baseline, if you will, of what people are doing? And it even took us a year and I think 14 months. It was so a total of 14 months, a year and 2 months to get to oh my God, we were better than the thing that people were using before for one protein, right? And for one property of that protein. And so Yeah. So year one was a lot of like, hold on to your butts. And I told our venture capitalists, this is going to be more on the adventure side of venture.
That's why it's called venture after all.
Exactly. Yeah. Yeah. This is not like early stage private equity as it sometimes is in Europe. But thank God that worked. And that really gave us a baseline of, hey, we can now go out into the market. And so phase two was sort of, okay, can we go from that technological hypothesis being correct to a value hypothesis? Can we actually apply this in an industrial context for something that's industrially relevant? Now, my earliest customers, they didn't believe this stuff. They're like, yeah, we've had like 2 decades of people telling us that computers were going to engineer biology. This is all pre-AlphaFold, and so they were even more skeptical than they were today, right? And so I actually had to pay our first 2 customers to try this. So they were like, okay, we're fine trying it. Then we'll make like a copy. We'll have more capacity in the experiments, but you'll have to pay for that extra capacity. And so we did that. And lo and behold, it worked a lot better than what they were using before. And that case study was sort of the moment where we could say, okay, we've proven the value hypothesis that this actually measurably provides, less cost and better product. And then that was also that was the moment where we also started building the application around it, sort of starting to make it more sort of scaled and robust and that kind of stuff. And that was the moment where we could start scaling it into larger application areas. And this was March of last year. And so it's been a really wild ride where, I don't know, I think last year, January, we had 5 molecules on the platform. I think we're now at like 47 or something in under a year. And so we're growing really fast. But, it was 2.5 years of like praying.
Yeah. Or the equivalent of praying.
Yes.
And now it works really well because you recently won the Adaptive Bio 24 challenge.
Mm-hmm.
Can you tell us what that challenge is first and how you won it? I mean, my understanding is that you won it actually quite decisively.
Yeah, I would agree with that. Maybe to contextualize first what our users are seeing, and then I'll go into the adaptive bio competition specifically. So I was referring earlier to, it's 10 to sometimes even north of $100 billion to develop one of these molecules, fully capitalized. What we're seeing with our customers right now is that between 1.5 up to 12x speed ups, where the middle is sort of 2 to 3x faster. And so that also means you can cut your cost by roughly that amount. And so a lot of our customers are quite excited about that. Now, where with Gemini or with, a human language model, you can evaluate them against big benchmarks. And there's a lot of good benchmarking data out there. In biology, it's very expensive to benchmark, right? Because you have to go into a laboratory and test all these things. Luckily, amazing Swiss biotech company, Adaptive, they're in Lausanne, right across from where you are right now. They're a contract researcher. So they're sort of like Amazon for laboratories, where you can like order a certain set of experiments on a particular sequence. And so they ran a competition where the goal was to come up with a better binder. And a binder is, in the drug context, something that grabs onto your target of interest. So, if you, in this case, EGFR, epidermal growth factor receptor. So this is a button on a cell surface that triggers growth. And obviously we don't like that in cancers. Like, we like to suppress growth as much as we can. And so, there are several products in the market that target this target. One of them is called Centuximab. It's marketed by Merck. I think it's grossing north of $1 billion in revenue annually. And so Adaptive was like, find a better binder against this target. So this is something that sort of grabs on more tightly. Now, why do you want that in a drug? Typically, it means it will hold on longer. So it's typically more efficacious. And if you can think of many proteins as sort of lock and key. And so if your key fits snugger into one thing, it's less likely to float around and accidentally try to fit in some other hole. And so what they call off-target effects. So where you accidentally grab onto something else and create a problem goes down. Binding affinity, which is how tightly it fits, is measured in nanomole, and lower is better, or femtomole or whatever, molar. The commercial product out there, I think, is about 9.2 nanomole affinity to the target. And we were able to generate a variant that was 1.2 nanomoles, so about 8 times tighter fit than the commercial product out there. And this was only 2 teams achieved this out of the 130 teams that participated in this competition. And our result was indeed the best result in that competition. What I personally find most cool about it is this was done by someone that at the time was even interning with us with no background in biology, but a background in computer science in a couple of hours using a piece of software. And so it kind of speaks to the democratization of these types of tools for people. Now, I need to also caveat this with having a better binder doesn't mean you have a fully developed drug, right? It's only one property that people care a lot about because it translates to efficacy often. But, there's lots of other things that you need to do in order to have a drug. Like it needs to be stable while expressing and sort of yada, yada, yada. You need to fully develop it first. But I think on this particular challenge, we did very much see that computers are quite good at this. And you can give it to a lot more, let's say, layman teams and get to much better results.
Yeah, oh, that's super exciting. So you mentioned you're now growing quite a bit. So what are your pain points at this point? Is it talent or data or compute or all of the above? And how do you predict that to change over time?
Yeah, for sure. I think there are a couple of places. So one, I would say organizational challenges. Second would be talent for sure. And thirdly, data always. In biology, there's never enough data, I think, in any ML application, but even more so in biology. I think, first of all, organizationally, a lot of biotech companies they need to spend a lot of money to try this, right? So let's say one round of experiments costs somewhere between 80,000 up to maybe more than $1 million. Sort of us coming in and being like, hey, why didn't you try this on your thing? They're like, yeah, well, show me that this works for my specific context. And so we're still in the early adopter stages, and there's a lot of evidence building going on. They're scientists. They're, I think, rightfully skeptical of ML techniques in general, and especially given the amount of hot air that's out there in the market generally. I don't think we'll cure all disease in the next 5 years. That's just bullshit. The second, and so organizational change is something, it's also quite emotional for a lot of scientists, right? They're like, wait, but I was supposed to, do science, right? And so, is this something we want? I think generally, procurement processes in large pharma companies take, forever. Like a year is not uncommon. And so how do you land with an account? How do you expand? How do you make sure that that works is one. Within that organization, second thing is data. Coming from Google, I would say biotech and pharma are very unsophisticated when it comes to their data infrastructure, right? It's really, and I'll not disclose, but I was allowed to use the quote from one of the pharma CIOs we work a lot with. He's like, well, Stef, it's spreadsheets all the way down. And so they can sort of think of, we're going to train all this cool ML, but there's really not a lot of data to train on because all the metadata is not there. It's locked into team drives. It's, et cetera, et cetera, et cetera. So data is a real issue. Thirdly, a lot of biology is, let's say, very multimodal. So like the types of molecules people are trying to develop, the types of experiments that they run, the types of problems that they want to solve are complex. And in biology, in contrast to tech, where a lot of people love to talk about all the things they work on, in biology, everything's IP and right? And so for me as a product guy coming from tech, it's like playing Red Alert. There's like fog of war everywhere, and you like drive to the other guy's base. You're like, oh my God, there's all this other stuff that we need to solve. And so you're even more building the plane as you're flying it. So that's the, let's say, organizational challenge of scaling. It's like features, data, and adoption, and skepticism, and I think rightful skepticism around the adoption. Second is talent. So I think a lot of people like applications like consumer apps where it's pretty clear if it works or not. It's really easy to build empathy with your end user because it could be you. That is harder in scientific fields, right? So whenever you join as an ML researcher or engineer at Cradle, you're going to go into a lab for 2 weeks and pipette and, learn about biology. There's a whole training program, and you kind of need to want to do that and learn this whole new domain before you can be useful in it for a while. So, and, finding that talent, but also training them, retaining them is challenging. It's a highly competitive market out there right now. I think the last thing is data. To your point, we spend a lot of money on our in-house laboratories just to make sure that we can make the cycle shorter. Like, for example, initially it took about 3 months to run around. It's now down to 2 weeks. We want to get it down to 1 week, because then an ML researcher can actually try a new hypothesis or a new method really fast instead of waiting a quarter, right? Where they literally have no memory of what they were doing a quarter ago. It's throughput. We've realized that a lot of our analytical chemistry methods, so the types of experiments that people run, are geared towards humans, which makes sense, right? Because we designed them for humans. But it turns out you can make experiments that are very good for computers, right? So, for example, there are only 22,000 proteins in the human body. And so I asked one of my co-founders, Harman, and we just try to bind to them all, right? Now, for a human, like billions on billions of interactions are very hard to let's say, internalize, right, and learn from. You can maybe cluster and do some data analysis, but really learning from that is hard. Turns out computers can learn from that data really well, right? So we're trying to rethink what kind of analytical chemistry can we do? How can we introspect biology at a very large scale in order to learn, the things that our customers want to do, and therefore allow them to go even faster and have even better molecules ultimately. So never a dull day.
Yeah, never a dull day. It doesn't sound like it. So I want to come back to something you said earlier, right? When we talked about fields of application, and notably, right, this is not just pharma, even though I guess that alone would be a super interesting field. But you recently, I think in March, published a report with other CEOs and founders in this space under the umbrella Advanced Biotech for Sustainability. Can you tell us a little bit more about what that is?
Yeah, for sure. And I think to your point, so pharmaceuticals is massive because humans are willing to pay a lot of money to not die. And so that industry is enormous. But we were alluding to it a little bit earlier. And so, McKinsey previously calculated for the cases where we currently use animals, or where we use plants, or where we use oil, so hydrocarbons, we could be using biology. Now, let me just rapid fire a whole bunch of applications that are quite helpful there. So take agriculture. In agriculture, we use a lot of herbicides and pesticides to get rid of, potato blight or whatever attacks your plant. Now, similar to pharma, a lot of people historically were using chemicals, things coming from oil. They are very toxic to the soil. They have all kinds of side effects when they get into our water. And so increasingly, companies that work in agriculture are using proteins to target things. Like, if I can find a way to just kill the potato blight without everything around the potato, that's awesome, right? And farmers like that too, because you get much better biology that way. You're seeing areas where we are using things that are bad for our environment, and we can move to a much more sustainable and less toxic and problematic alternative. Second category is places where we are currently using oil. Like, for example, in materials, we use a lot of oil for plastics, for fibers. Like, if you're wearing nylon, that's made from oil ultimately, and we would like to use less of that for two reasons. One, it's bad to have lots of oil, but it's also bad to throw that stuff back into nature, because nature doesn't know how to break down a lot of plastics, right? And so we know, and biology is very good at making incredibly strong fibers. Like, look at your random house spider this afternoon and just think about how much weight is on that little line. So if we can harness those types of things, we can improve. Same for, let's say, food products, right? Like, we currently use cows to produce effectively water, whey protein, and fat. We call it milk, right? They're not a very efficient way of producing milk, right? If we just want to make whey protein, we could make whey protein. Okay, so let's assume 60% of the things that we currently consume could be made with biology. How does that look? And I think this will be a big area of society in the future. It's like if you study like agriculture, you could probably pick the cattle, the greenhouse, and the fermentation major, right? And so this works similar to brewing beer. So if you make beer, you take water and sugar and some grains of your liking. You throw a microbe in, and that microbe will start turning the sugar into alcohol. Now, very similarly, we can sort of take that little microbe, make changes to how it works, and make it produce other things, such as whey protein, like milk, such as materials, such as chemicals, right? And so this ranges from beauty products where, you want things that are maybe not alcohol-based to disinfect your hands, right? You could use biology there. It can be the detergents that you use. It can be the toothpaste that you use every day. All these products, and they are partially already made with biology, but more of them could be made with biology in the future. What does that do? Less land use, right? Significantly less land use. We use a significant portion of the earth to have cattle run around, right? Second, you can make these products with earth abundant materials, right? So we're not going to run out of water. We're not going to run out of sugar because we evolved in a context where that was abundant, right? And so you can take that locally. You don't have to go to Saudi Arabia or, a rainforest to get palm oil. You can make that locally. And so you have a lot more independence in terms of your supply chains. And given what's happening in the world, that might not be a bad idea right now. And then thirdly, biology ultimately finds the lowest energy way of doing very complicated chemistry, right? And so you can make things that are much cheaper energetically than what it would take otherwise. So less space, less water, less energy, no harmful side effects.
So, just to back that up with some of the statistics you give in this report, right? You basically say by 2040, this tech could reduce CO2 equivalent emissions of 5% of global emissions, free up 2 to 4 million square kilometers of land. That's about the size of India, and create about $1 trillion in economic value, about the size of the Swiss economy. So that sounds very compelling. What needs to happen to sort of turn this vision into reality? We just wait and it happens on its own, or can we accelerate this?
There's a couple of things. So I would say societal acceptance of these products is something that is improving, but is still tricky, right? So we don't like things that are engineered biology, although, we literally inject half of the Swiss population injected themselves with coding RNA like a couple of years ago. But generally, societal acceptance of these types of things, I think even if you just take the subset of it's made with engineered biology, but the end product is equivalent to, something that's out there, we could already go quite fast. The unfortunate sort of side effect of that is that a lot of our legislation is quite restrictive when it comes to genetically engineering anything. In the US, that's a lot easier. In China and in other places, that's a lot easier. I'm a big fan of having safe and good stuff. And there are lots of easy ways in which we can do that, because we also have to do that for whatever random protein you find in the Borneo jungle, right? Today, you can't just put that in a food product. You need to, establish whether that's safe or not. But this requires some change of the law. Then I think in terms of, let's say, technologically bringing this to reality, there's two parts that need to be fixed. One is, or three parts really. One is the protein engineering bit, which we are working on. Second is strain engineering. So how do you make the cell produce the thing where you have maybe many proteins working together? That's still an unsolved problem. It's a company somebody should start, because it's a really cool problem, but not solved. And then the last bit is scaling it up to, hectoliter fermentation tanks and making that process easier and more capital efficient. Like I said, if you look at companies like Novozymes or IFF that are customers of ours, they already produce a lot of this stuff at scale. It's just the types of things that you could be producing in terms of price per kilo could come down, and then you get a lot of these environmental benefits as well.
Hmm. So it's interesting that you mentioned that also from a societal perspective, because I mean, all the use cases you describe are amazing and they're positive. Someone listening to that could think, this tech, I mean, that all sounds great, but could probably also then be used to do bad stuff with it, as always, right? What's your thinking about this? I mean, is that just basically something to take care of with regulatory, or are there technical guardrails you can put into the process of preventing that from happening?
Yeah, certainly. So I think safety is of the utmost importance, and I would want to distinguish here proteins from full cellular systems, right? So proteins don't self-replicate, whereas cells do, and so the worry of, okay, something runs out into the world and starts multiplying itself. You've commented on that in the past. That is actually way more dangerous than proteins themselves, because again, they don't mate and make more of themselves. That said, safety is still critically important. There's a few things that you can do. One is there are lots of things we know to be bad already, and I think biology and biotech companies have actually done a quite good job regulating that. So if you order DNA, so for people that don't work in biotech, the way that you do this stuff is you make a sequence, you then call companies that know how to synthesize DNA and make you the little long strand. They have checks on what you can and cannot make. And so, for example, toxins, carcinogenics, those types of things are known and marked. And if you want to use them, you need special permission in order to even order something that you could express and physically instantiate in a laboratory. So that's one. Second, and obviously there is a ton of already very dangerous stuff out there, right? I mean, you can find all the sequences of all the scary things in databases, and you can just order them, and you could decide to do bad stuff with that. So anyway, second is we need to make sure that in software tools like ours, we have safeguards against misuse. And so right now, whenever a company onboards with Cradle, we check, do we trust these people? Yes or no. If you ever read on the weekend Cradle's terms of service, there's specific applications that are disallowed using Cradle for. And so, you need to, as a software developer, have strong controls on what can and cannot be done, and you need to make a decision on what you allow and disallow use for. So that's I think the short of it. Then before products, and this is sort of all in the research and development context, then before products make it to market, we have established protocols for this, right? So it's not like people were not engineering biology before. The only ways in which they were doing it was more random, right? And so I think with these AI systems, we have way stronger guarantees that things might not go wrong than we had before, because these systems are much better at introspecting. Are there certain side effects? Might this be toxic? Might this create an unwanted immune response? Those types of things. And then, we have a process at, the Federal Drug Authority, for example, in the US, the European Medicines Agency, but also for food. There are strict controls on what you can and cannot formulate, and it needs to adhere to strong safety standards. And I'm a big fan of that. So we should continue to do that for AI generated, as well as random stuff people find in the jungle, or, shine some UV light on to introduce random mutations.
Yeah. Good. So let's switch gears here a little bit and talk about the fact that you are, let's say, a European company. Actually, so you're in Zurich, obviously, but you're also in the Netherlands. Is that right?
Yeah.
Okay. And so, you're actually very outspoken about the fact that building a company like yours is challenging in Europe. I follow you on LinkedIn, which is something I recommend to anyone. And you had a post on LinkedIn recently. I mean, you post a lot, and it's always interesting, but one of them went viral. And if I can, I just like to quote from it, because I think you really sort of, kicked off a super interesting discussion. I mean, basically, you said, quote, "I tried to hire a superstar machine learning engineer when I told him it would take me 8 weeks. to get the offer ready because of Dutch visa processes, he laughed and started working for OpenAI a few weeks later. This is one of the very real, very practical examples that make operating in Europe 10% harder every single day. It also took us 3 months to open up a bank account. It cost us 70k euros on notary and legal fees and weeks of work to start a company. In the US, it costs $500, and your incorporation documents are ready to go through Stripe Atlas. I'm optimistic. You had other texts as well, but I'm not going to read the whole post. But you close it by saying I'm optimistic because there are a lot of smart people in national and European governments trying to solve this issue. But if we want to continue to be competitive in the spaces that we still have a competitive edge like bio, we need to get moving fast, exclamation mark. So my first question is, like, how can you actually still be optimistic given these experiences? So I'd like to know that. And, there is a sense in your post that, something seems to be changing. And when, one reads your other LinkedIn posts, is that really true? I mean, can you give us a bit of insight of your own experience there, what you currently see in Europe and what should happen and at the speed at which it's happening?
So let me first contextualize it at the macro level. So a lot of the technologies that were developed over the last decade were net new. So the iPhone was net new. Big data was net new. Social networks were net new. They created new pieces of economy. My worry for Europe is that a lot of the newer technologies like AI, they're coming for existing incumbent industries such as pharma. And we've seen this play out in Germany with Volkswagen and Porsche. And I really want to avoid that happening to more European industries over the next 5 to 10 years as this new wave is coming up. So what do you need for a company? You need a bunch of smart people. You need money. And then you need everybody to just get out of the way. That's really it. And so anything that inhibits companies from running or hiring or attracting capital or those types of things, even if it's, it sounds individually reasonable, let's say, it's not unreasonable to wait eight weeks for a visa. That is true. It's not unreasonable. But if you sum everything up, everything is just a lot slower because all of these things compound. And so I think in Europe, we've stopped sort of focusing on the user and which is the companies in this case, and we've over-focused on sort of potential risks and managing those risks. So for example, I don't think random idiots should get a bank account. I also think that I had banked for 8 years with that bank, and I'm pretty sure that I'm legit, right? And so in this case, KYC legislation probably went a little bit too far because without a bank account, you cannot transact, right? Like you cannot operate a business without a bank account. And so if you put a tax of 3 months on every European, in this case, Dutch business, that is somewhat more complicated than your standard business, you're going to be delaying every company by 3 months, right? And again, right, there might be good reasons for wanting to make sure that people are legit, but I think this is a little bit overdone. And so why am I optimistic about Europe? We have a shit ton of money. Right? We have an insanely high and deep talent pool. We've been kicked in the nuts for the last few centuries every time, and we always surfaced. And I'm not worried about us not being able to do this. I do think bureaucrats need to get out of the way, right? And they need to stop making things too complicated. Now, I'm not saying blanket deregulation, but be very smart about what you do. So for example, a bank account, you can open the bank account and start your due diligence. You can say, hey, don't transact over this amount and don't transact out of the borders, for example, to limit any sort of risk. And then we'll give you a final answer 4 weeks from now. Right? Or similarly with, immigration laws, and I know this to be true, for example, here in Zurich, for some of the large companies, they get a quota, right? They get to hire a certain amount of people from outside of the EU, and no questions asked, they can just issue a contract. Why is that only available to large companies and not to small ones, right? And so these are not hard things to fix, right? And I think, and the reason why I'm optimistic is that is now being recognized a little bit more. Now, whether or not we're going to see mostly window dressing or actually concrete action, we'll see. But let's say 2 years ago, nobody cared. Right? And I think with the Orange Man in the White House, a lot of people seem to be caring a lot more than 2 years ago. And I am very long European ingenuity, and we have very deep capital markets. So we should be able to get ourselves out. If we don't, it's of our own doing, right? And then we become an open air museum and just manage people's money.
Yeah, which doesn't sound like a great future. So how could universities help? I know you had some pain points there as well. Um, can you tell us a bit more about that, and then sort of tell us what you think needs to happen?
Yeah, Switzerland does incredibly well. And so I think both federal, or well, there's more federal institutes, but both EPFL and ETH are quite good at this. I think ETH, as a publicly funded institution, is on par with global private universities in places like the United Kingdom and the US in terms of numbers of spinouts that get produced, total market capitalization of these spinouts, and the number of people that join them. I think there could be more depth, as in we're always very socialist in Europe in general, so everybody should get a little bit of money. I think it would probably be good if, let's say, we want to win in robotics. Right now, we have Antibiotics, we have Ethan, we have a bunch of really awesome startups here in Switzerland. We have ABB, and we could really win. If you look at like spatial reasoning in AI, like the iPhone's AI for the camera is made here in Zurich. Disney's 3D labs are here in Zurich. The Facebook, what's it called? The little thing you put on your head, the 3D thing, that's made here in.
Mm-hmm.
We're one of the best when it comes to spatial reasoning.
Yeah.
There is no reason why we couldn't kick Elon Musk's ass and build by far the best robot that actually works and isn't just remote operated by humans, right? But we do need to decide that we want to win. And so we should not be complacent. And I think if we add a few deeper bets on things where we believe we want to strategically win, we could. But we just need to move.
But there, concretely, what is it that that you would tell universities to do or not do?
I think for honestly, the federal institutes keep doing what you're doing. So be fast, don't take too much equity, build that whole startup ecosystem around it. I literally had my alma mater, University of Groningen, visit here last week to do a tour of ETH and InnoSuisse, and I think that's all done very well. I do think we're a little bit too much. Here's like 2 postdocs that get 200K Swiss francs. There's not the, like what Americans do and Chinese do. It's like, we want a factory that can build the same machines as ASML next year. And they actually do it, right?
Yeah.
And so I think we could do that here as well. We could say robotics and precision industry is something we want to retain a lead in. Go, right? We're going to deploy 800 million Swiss francs, and we're going to put it in 5 companies.
Mm-hmm.
Something like that. So like specific bets on areas that we care about. And there are a lot of good things, right? We have a large food industry, we have a large banking industry, we have a large biotech industry, we have a large robotics industry. Pick the top 3 and go.
Yeah. So, okay, how could that happen? I mean, I totally subscribe to the vision. The challenge at the moment is, right, that the political situation is simply such that, there isn't just gonna, there's not gonna be much more money for those actors from the government, just because of all the debt, laws and so on.
Yep.
So now, of course, we can just wait around and say, well, we'll just wait for better days. But there must also be other ideas, right? So, I mean, okay, let's imagine for a second, right? You suddenly would have the ear of a few people or institutions with serious resources, and they want to back an exciting vision to put really Switzerland there as a leader. So, and of course, right, there's money involved, so they also want to make smart investments. What would you actually do, right, if you sort of would get that kind of blank check, of a couple $100 million?
I do always love it when sort of you're in Switzerland and the Swiss are like, oh, we don't have enough money, right? It's like the most fortified balance sheet of any government in the world.
Yeah.
Like, sure.
Well, maybe that's why, right?
I think we're deciding there is no money, but I don't think that's really true. Anyway, that's maybe for a different discussion. The question is, what do you want for your kids, right? And so I don't think you can wait and see. I think you need to act. You can act in many ways. So you can have the government act and decide on whether the rules that are made today are still relevant given the geopolitical context that we're in. But separate discussion for the Nationalrat, I guess. The second thing you can do, and I think Switzerland is actually quite good in public-private collaboration here. And so, I've seen many initiatives that are like sponsored by UBS and some of the large institutions here that are there to promote Switzerland and Swiss activity. And so I think you could definitely build a coalition of the willing to do something like this and make a few big bets. I don't see why like the Norwegians and the Singaporeans, there couldn't be the Swiss Temasek or Swiss Foreign Wealth Fund that sort of deploys money specifically geared towards things that should be built in Switzerland. We have incredibly wealthy families for which a lot of this stuff would be an absolute rounding error on their annual proceeds on their assets. And so I think money can be found. If we cannot find money in Switzerland, it's kind of funny. It's mostly willpower, not the existence of money. Right? Things that you can do. I think what I think you should do is look at where do people invest that are highly that are very good at this game. And so, and this is really something I always find somewhat surprising in Europe as well. They don't make venture capitalists part of the European Commission, right? Because they don't have a lot of experience. But for some reason, you can spend billions of dollars making investments in startups, right? That is a bit weird, right? So practically what you could do is something like, hey, we'll take top few hundred venture capitalists in the world. We'll look at what kind of bets they're making in industries that we deem strategically relevant to Switzerland. And we're going to, give them the multiplier. So if you're able to raise $100 million from Sequoia, great. $400 million. Go. Compete with OpenAI. Because that's the type of money that's currently floating around, right? And what's being deployed in the United States, what's being deployed by Saudi Arabia, what's being deployed by China, right? And if we want to compete, we need to build some winners, not just distribute, if that makes sense.
Yeah, no, that makes sense.
I think scientifically, there's something else to be done, which is, what's happening in the United States, and I'm obviously in biotech, is quite remarkable, right? So on the early research, we're seeing, 40 to 60% slashes in funding for biotechnological research, which literally means that even Nobel laureates are now laying off people that are highly talented. I think secondly, we're seeing, China is not a place where you can do business anymore. We're seeing drug prices being pushed down. We're seeing tariffs being imposed. And so biotech and pharma, which is a large contributor to Switzerland's GDP, is under a lot of pressure right now. But it's also an opportunity because a lot of innovation from biotech and biopharma is coming from, the Broad Institute at Harvard, from the Institute of Protein Engineering in the US. And what we could do is say, hey, here are a couple of concentrated bets. We're going to take biotech robots. We're going to take, these four things. We want to win, and we're just going to allow these institutions to open up shop at EPFL and ETH. And we'll take the people that are being laid off. We'll build deep connections. Worst case scenario, if the United States academic system implodes in the next 8 years, we have some great people. And I'm hopeful that this will happen. That doesn't happen, and there's some return to normalness in the US. We've built incredibly deep connections with some of the best research institutions in biotech, in robotics, in whatever the areas are. This is really an opportunity and not a moment to be sort of penny wise, pound foolish.
Yeah. And I think there could be really creative new models, right, of monetizing this. Because again, unfortunately, I think the money for that is not going to come from the government at this stage. Even if they would want to, they just cannot. It's all a question of will, but
there's no parliament that can approve laws. Is that what you're saying?
Yeah, no. But the political realities, right, will just not make that happen within the next 6 months or even 12 months or even longer. But at the same time, there are people who could foot such a bill. But that would need to go along, I think, with an interesting model of potential returns, right? Where you could say, okay, we could create such a research institution, but then at the same time, It's going to be highly competitive and highly collaborative also with the existing system. But at the same time, the people who funded this in the first place, of course, get sort of first shot at the startups that are coming out of this.
Yeah, or you have some form of, early investor ticket that you do and you retain some equity. Like, especially if you let experts pick, right? So, I think Sequoia even has like a 60% write-off rate, but they also have some of the best returns in the industry. If you get to co-invest with people that know consistently how to give you good returns, it's even more likely that it's not something you're going to throw down the drain, but where you actually make money over time. And if there's one thing that I've learned as a sort of living here is that the Swiss love opportunity, right? They'll jump on things if they can make some money. So I think that's a model we could try, and I think it requires a certain focusedness of investment and excellence. Yeah, for sure.
Yeah. Well, super interesting. Let's talk more about this later on. I have a last question I want to ask you, and this is particularly dedicated to the students or just generally, right, the younger generation. So you mentioned this right now a couple of times. Okay, it's actually super exciting area now in biology, biotech, and it's not just pharma. It goes into sustainability and so on. And the kind of skills that are needed are also different from what they would have been, right, if we had this conversation 10 years ago. So what's your recommendation, right, for someone who says, okay, I want to build at that interface, right? What should I do? What skills do I need? What kind of projects, what kind of experiences should I seek out now?
Yeah, for sure. I mean, it's a complicated question, right? Because it depends on many different things. But I would say, even though AI is getting better and yada, 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 I think fundamental understanding of physics, math, and programming will remain helpful because people need to actually design those ML systems. And at a minimum, do that. I think biology is a more complicated field in the sense that, we didn't come up with biology ourselves. And so we're still learning a lot about it. It's kind of one of the things I love about biology is that you kind of have to poke it first to make an instrument to then go introspect it again. And so, biology is an area that I'm super bullish on. There are plethora of problems in biology, and we talked about some of them. It's like, figuring out from a large group of patients why they are sick, like what's mechanism of disease, like major issue with the coming of better machine learning models. Will learn about why disease and why aging and why those types of things occur, and we'll probably be able to influence them in more positive ways. I think in experimentation, right, there's so many experiments and instruments that could be built that aren't there yet to sort of introspect biology and make it more engineerable. I think for those that are not in biology but are in computer science, let's say the software stack for people in this industry is absolutely garbage, right? And so there's so many things to build, from like interfaces to lab equipment and organizing their data to how do you analyze and introspect? How do you go through large corpuses of papers and summarize the relevant bits? How do you go through large databases of patient records and figure out what's happening, in non-biology or in non-medical applications? Like, what are all the cool like syntheses, like chemistries that we could be making with biology that we're not making right now that could be super valuable to people? How do we build reactors like that can produce a lot of material but are cheap to operate and build? In tech, when I was at Google, at some point it felt like, okay, we have like 15 chat apps or something, and we're starting to get to the point where most things have been made. And honestly, I have the same feeling of some of the large language model stuff. It's like, I think we have enough marketing chatbots at this point, right? Like, go build something useful and go do it in biology.
Okay, well, Steph, this was great. Thank you so much. I know you must be super busy building this rocket ship that Cradle is. Before you leave, where can people find out more? So on the one hand, about you, I mentioned LinkedIn. Is that sort of the best place to get exposed to your views?
The only place.
The only place, okay. And for those who want to find out more about Cradle?
Best thing is to go to cradle.bio.
Cradle.bio. All right, and we'll link to that as well. Good. Well, thank you so much. This was terrific.